Good evening. Good evening. Let me see how many people are. Okay, I have 12 people here. We are starting once it gets to 20. Can you hear me? If you can hear me, type yes in the box. Okay. Okay. We have 14 people. I'm waiting for. You guys have to be complete though. The other class, all of them were here. Hmm? $17.24. Okay, perfect. Okay, hello. Past my bedtime, but I'll stay up as long as I can. Ha. Better go and get a coffee, and drink, and be awake. Because we are crashing. There's something they call crash course. This is a crash course. You understand? This is a crash course. That's why I'm surprised that everybody is not here, but it's fine. Their choice. Okay, how are you guys doing? Good? Yes. Even if you don't tell me, I know you're doing good. Whether you tell me or you don't tell me, I know you're doing good. So, I guess they are just getting the message. So, but I will crash your FDR. Please don't do. Please don't crash on. You're very funny. Okay, so let's go to business. I'll give you guys the rule. The rule is, yeah, we can start. Yeah, it's fine. I'm going to be very fast. That's number one rule. I fly because I really want to finish up this in 2 hours 30 minutes, which may not be possible. Do you understand? But it's fine. Whenever you want to sleep, you can go. That's fine. I'm going to start with genetics, then we do tissues, and we end it with skin. All right. The first thing I'm going to do is I'll show you the slide, then I'll just touch 1 or 2 places in the slide, and I fly into questions. This is not teaching class. This is last minute review. That's why that's what I always call it. It's last flight. Okay, it's last minute review. All right. So don't expect me to start teaching things that I've already taught or things that you can watch in the videos, because the videos is there for you to get clearer explanation. All right. So this is just hello, welcome. This is quick, fast pace. Okay. So hold on. Just a minute. Just a minute. There's somebody that is not supposed to, that didn't get this email, and I wanted to send the person a message. Okay. Okay, so I'm going to share my screen now. If you can see my screen, I need you to type yes. Oh, so the rules of engagement is everybody must comment. When I give a question, either A, B, C, D, or E, everybody must answer that question. If you don't know the answer to that question, you don't need to follow the crowd. All right, good. You guys are active. I need activity. I just finished mathematics, 3 hours mathematics. with the mathematics students like some minutes ago. That's why I delayed in sending you this stuff. I was supposed to end by 8 o'clock, but they pushed me till almost 9. Yeah, so, and they were very active. So I expect you guys to be more active than they are. Okay, so what I need you guys to do, everybody, if I ask a question in the box, I'm throwing a lot of questions. Attempt it. If you get it wrong, fine. If you don't know the answer, write 911 or emergency or code blue so that we know that you need clearer explanation on it. All right? That's our code. That's what we work with this night. So the faster, the better. So I'm happy that you guys are seeing my screen. I'm going to power this screen for this course. But the problem with this screen I'm projecting is that it's always making me lag. All right? So if I'm lagging too much, put it in the box. But immediately I finish touching everything I need to touch, we jump into questions. So I'm taking 10 to 15 minutes to touch everything I need to touch right now. All right? Okay. But if I'm lagging too much, please tell me in the box so that I will look for an alternative method. So the first thing we are going to look at is genetics. Genetics is extremely important, all right? You know why genetics is important. I don't need to overstate it. I'm not going to waste time. I'm flying. If you want to fly with me, you are welcome, right? These are the things you're expected to know. Okay, look at this. So we have DNA, right? Which is majorly the foundation of genetics. You have to understand the DNA structure and its function. Important that you understand it. What is DNA? Long thread-like molecule with uniform diameter and varied lengths. Hold on. Sorry, just a minute. Okay, sorry. Trying to be sure of something before I continue. I'm getting ready. Okay, so the DNA, we know what the DNA is already. I don't need to overstate it. We have 46 DNA molecules, that's chromosomes in the nucleus of most human cells, all right? Now, DNA and other nucleic acids are what? Polymers. What are they polymers of? What are their monomers? Nucleotide. They will ask you this question in your exam. You need to know that they are what? Nucleotides. And they will ask you, what is nucleotide composed of? Sugar, phosphate base. No. Sugar molecule, phosphate backbone, and nitrogenous what? Base. All right? So I'm not repeating it. And now, what do you need to understand? For DNA, the sugar is what? Deoxyribose. For RNA, the sugar is ribose. The DNA and RNA has a phosphate. DNA and RNA has nitrogenous base. All right? But nitrogenous base is divided into purine and pyrimidines. Purines are double ring. It sounds small, so it's double. The one that sounds big is single. Purines, double ring. Pyrimidines, single ring. Now, purines are adenine and what? Guanine. Pyrimidine is where the problem is. We have CTU, cytosine thiamine for DNA, and we have what? Cytosine uracil for RNA. So if they ask you, what are the pyrimidine rings? The three of them are pyrimidine rings. They may tell you, all the following are pyrimidine rings except this. I'm going to give you a question. Just relax. Relax yourself. Don't write. Don't write, please. I just need you to just focus. Get glued on the system, all right? And get activated for me, all right? So this is the diagram. I believe that all of you can see it. But if you can't see it, always tell me you can't see it. We have adenine, we have the guanine. This is adenine, this is guanine. The two of them are purine rings. You see, purines. You see, they have double ring, double finger rings. But the pyrimidines are single ring. You need to know which ones are pyrimidines and which ones are purine. They will ask you this in the exam. By now, you should be singing it. You should be in the bedroom and singing, with a good voice or an ugly voice. Purines, adenine and guanine, pyrimidines, cytosine, thiamine, and uracil. That's what you need to do. If you want, I can come and sing for you. No problem. All right. So let's move. Now, in terms of pairing, you need to know that adenine pairs with thiamine for DNA, but for RNA, adenine pairs with what? Uracil. because RNA does not have thymine. Then we have cytosine and guanine. And they will ask you which one has 2 bonds, 2 hydrogen bonds? We have A and T. Adenine and thymine has 2 hydrogen bonds. Cytosine and guanine has 3 hydrogen bonds. CG, 3. AT, 2. CG, 3. Put it in your head. I'm flying. Sorry. You need to know what gene is. You need to know what genome is. Huh? Gene is a segment of the DNA for synthesis of a specific protein. You need to know what genome is. That genome is, all the genes of one person. All your gene is known as what? Genome. All right? Because they will ask you that question and they want to confuse you. Don't allow them to confuse you. You know what chromatin is? They are fine filaments of DNA molecules that are complex with what? Proteins. They are tied with proteins. They occur as 46 chromosomes in most cells. So when they are activated, they become chromosomes. When they are not activated, they become chromatin. I told you this before. Okay? So this is a chromatin structure. I'm jumping it. We have the histones that the proteins that helps to pack DNA as chromatin. All right? Okay, good. Genes can be turned off or on. Please remember that during development, chromosomes migrate so that genes on different chromosomes can partner to bring about what developmental changes in the cell. All right. So sometimes genes can be off. All right. Why sometimes they can be on? Make sense? Now, another thing you need to know is what kinetochores are. Kinetochores are. They are like plaques. You know what plaques are? Like people that are having arterial blockage. Arterial blockage. You see a plaque will fall out and go pew around the body and come and block one of the little arteries. And before you know, especially the one that goes to the head, leading towards stroke. Because there's no blood supply. It cuts off blood supply momentarily to the brain. So, but that's by the way. I just want you to understand what kinetochores are. They are just plaques at the side of around the middle of a chromatid. But the middle of chromatid is what? Centromere. This diagram is extremely important. You must know this diagram in your head. You should close your eyes and draw it. If you notice, you see the kinetochores at the sides. They are the proteins at the sides. Then the middle is what? The centromere. These two things are sister what? Chromatids. Sister what? Chromatids. Make sense? They have not been activated. That's why they are called what? Chromatids. Okay, let's fly. Now, talking about the RNA, we have 3. I've given you the difference between DNA and RNA. Major difference is that DNA has deoxyribose sugar. RNA has what? Ribose sugar. All right? Now, there are 3 different types of RNA. We have the messenger RNA. We have waste trainer for the chromosome. I love you too. Thank you so much. Anybody that said that. Now, the 3 types of RNA, we have the messenger RNA, we have the ribosomal RNA, we have the tRNA, transfer RNA. You need to know what they do, all right? You need to know the differences between each and every one of them. But before we do that, let's talk about the gene. What is gene? We mentioned it before. It's just information, all right? Information. We already mentioned genome. How many genomes do we have? All the DNA in 123 chromosome set is what? 3.1 billion nucleotides. That's 3.1 billion. They will ask you this question. You know that figure, 3.1 billion. Billion dollars. Billion nucleotide pairs. Remember it. What is a nucleotide? We mentioned the 3 things that nucleotide contain, which is what? The sugar, the phosphate, and what? The nitrogenous base. Genomics is just talking about the whole thing, which is study. Study. Thank you so much. Study whole genome and how its genes and non-coding DNA interact to affect structure and function of the whole world organism. All right? Okay, this was talking about Homo sapiens. I don't care about Homo sapiens. Okay, so talking about application. We are still talking about genetic code. Now, one thing you must know is that genetic code they exist in 3 pairs. We call it what? Codon. Codon is the 3 base sequence in messenger RNA. We call it base triplets. I need you to think about it. If I tell you, that somebody has 50 base triplets, so how many chromosomes will it be? 50 times 3, 150. Know the mathematics because you will see a question like that in the exam. All right? Thank you so much. Somebody is smiling and somebody is very happy this night. Somebody is excited. Somebody is, let me see, how many people we have here? Oh, wow, 30 people. Perfect. So let me accelerate as usual. Now, you must know start codon and you must know stop codon. Start codon is AUG, August. I was born in August 25th, all right? So remember, use me to remember the start codon. My life started in August, AUG. Now the stop codon, please don't associate it with me, all right? We have the UAG, we have the UGA, and we have the UAA. Put it in your head. Close your eyes and say it right now. Don't close your eyes and sleep, oh. Just close your eyes and say it. Because some people are looking for reasons. They say, professor said that they should close their eyes. Now, let's talk about transcription, translation, and replication. Guys, I've described this thing a lot of times in my video, so go and look it up. All right? Go and look it up. Transcription is copying, photocopying of the DNA strand. Translation is what's making the protein. Sorry, transcription is writing the copying DNA to RNA, messenger RNA, that's sticky notes. Replication is photocopying the DNA. Translation is making the protein, all right? And also, you need to know these enzymes, RNA polymerase, DNA polymerase, helicase. What does helicase do? Helicase unzips it. It unzips it. The DNA polymerase what? Replicates, helps in replication. It copies each arm. Now, the RNA polymerase what? Transcribes, yes, helicase unzips. RNA polymerase transcribes the copied arm into RNA, messenger RNA. So you need to know the function of DNA polymerase, RNA polymerase, helicase, and ligase. Ligase does opposite of what helicase do. Helicase zips unzips, all right? While ligase zips. Those are the four you need to know. Trust me. If you know these four, you are good to go. And you need to know where to add the base pairs. I'll give you a question on that. Let me not stress on it right now. You need to know where to add the base pairs, all right? So we already discussed this. Alternative splicing is when you use one gene to make a lot of proteins. This is an example. You see, look at this diagram very well. In case of the ask you this diagram exam, look at this diagram very well. If you are looking at my screen right now, what do you see? You see, this is the DNA, right? This is the DNA. This part is where the DNA replication happens, all right? The DNA unzips by helicase, DNA polymerase replicates it. When it replicates it, what happens? It will be transcribed to pre-mRNA. So this position is what? Transcription. The first position is replication. The second position is transcription. It has been transcribed from DNA to messenger RNA by the RNA polymerase, all right? Then after that, we do what? Splicing, cutting. We cut it. mRNA1, mRNA2, mRNA3. That's splicing. All right? Alternative splicing. Then after that, they use it to make what? Protein. This is translation. You need to understand this sequence. All right? This is what they're explaining here. I need to know where. Where. You understand? This translation happens where? It happens in the cytoplasm. All right? Happens outside the nucleus. It happens in cytoplasm, specifically on the ribosome. And the ribosome is where? The ribosome is either walking around freely, or the ribosome is on the endoplasmic reticulum, the rough. The ribosome makes it rough. Then after that, the food moves to where? The Golgi for arrangement and packaging. All right? Very important that you know. I'm jumping all these things because we've already discussed them. Yeah, nucleus for, yes, replication and transcription happens in the nucleus. Yes, replication and transcription happens in the nucleus. All right. Cytoplasm for translation. Perfect. Thank you so much. Okay. Let me move. This is everything we just discussed. Remember chaperone proteins. Chaperone proteins are very, very important. They are these old proteins. You know, in the clinic, you see these old nurses. They will be telling you, if you see the kind ones among them, they will tell you, no, don't do this here, do this. They are helping you. They are teaching you. That's what chaperone protein does. All right. Chaperone protein are those older proteins that pick up the new proteins and guide their folding into what? Proper shapes. Without them, they can misfold. And when they misfold, it becomes a problem. All right? You talk about that when you get to pathology. So this is where everything starts in the nucleus, right? Replication happens in the nucleus. Then transcription happens in the nucleus. Messenger RNA moves now to the kitchen, which is the ribosome. Where is the ribosome found? On the endoplasmic reticulum. Then, or freely moving. After that, when they finish cooking, they bring it to, they package it to where? The Golgi. The Golgi does what? Something we call what? Post-modification. Post-translational modification. All right? So that's what happens here. That's what they are describing here. I'm trying to be very fast. I need to finish up and let's jump into the questions. I don't want to spend too much time, all right? But if you have any question, please drop it for me. Feel free. Feel free to drop it for me. Anything I don't explain, we'll catch it in the question. Now, mind you, cells synthesize glycogen, fats, steroid, phospholipids, pigments, and other compounds. They don't have any gene, all right? There is no gene to synthesize these compounds. They may ask you in the exam, the following does not have gene to synthesize them. Remember those things that I mentioned in bold. That's the glycogen, fat, steroid, phospholipids, pigments, and other compounds, all right? So these things, because they don't have any gene for them, what happens? The cell will produce what? Enzyme, and enzymes will now make them synthesize these things. It's as simple as that. So it's indirect production. Indirect what? Production. All right? So that's that. Now look at the DNA replication. All right? That's it, and cell cycle. Extremely important for you to know. Extremely important for you to know. DNA replication and cell cycle. These are everything that happens. These are everything that happens. We've already discussed them. Please, this diagram, use it to do your screensaver. It's very important, all right? You see, helicase, this is helicase, unzipping these guys. This is DNA polymerase. If you look at it, this is just one. That's the helicase, the original one, right? The DNA polymerase starts creating it, starts creating. It becomes double, starts creating. And after that, the DNA ligase is one that joins everything back up. All right? So you need to know the function of the helicase, polymerase, ligase, and what? RNA polymerase. Remember that. Yes, ligase joins everything back up. Okay. There are mistakes sometimes, yes, which can lead to mutation. Okay, let's talk about cell cycle, guys. Very important. Cell cycle. We have interphase having three subphases, all right? Interphase. Have G1S, G2. Then we have the mitotic phase, which has what? Pass my anatomy test, PMATs, that's prophase, metaphase, anaphase, and telophase. So PMATs. I pass my anatomy test, but there's one part that's remaining, which is cytokinesis. The cytokinesis always overlaps with telophase. You see this thing I mentioned? Put it in your head subconsciously. The cytokinesis overlaps with what? Telophase. All right? Yes. Cytokinesis overlaps with telophase. So we have I passed my interphase, passed prophase, my metaphase, anatomy, anaphase, test, telophase, completely, cytokinesis. So this is the diagram. You see this diagram? Very, very important. Thank you so much. Very, very, very important. All right. This is the three of them. I told you that this thing is when the metabolic activities, they are warming up. We are preparing. That's G1. Then this is F1, S1. What happens in S? This S phase, synthesis phase, S for synthesis. It is DNA replication. Mind you, it is DNA replication. After DNA has replicated, they go to the second stage, which is the growth and preparation. Everybody's preparation. Oh, we want to separate this class. Oh, we are so happy. DNA replication. We brought you a new anatomy teacher. Apart from Dr. Ray, Dr. Nchebe, we brought another professor. Oh, okay, that's replication. Then everybody's now arranging their bags. From there, we go to prophase. That is the D-day, all right, for that division. Prophase, the day for the division, everybody, are you guys ready for division? Yes, that's prophase. Then you go to metaphase, M for M, metaphase for middle, all right? Everybody come to the middle. Once you see them lining up according to their height in the middle, that is metaphase. Then after that, you talk about division. So that division starts with what? Anaphase. Mind you, they will ask you this question. Then after that, they move to the extreme of the pose. That is what? Telophase. So if you look at this diagram, you see this red, this thick red. It overlaps with both anaphase and telophase, but the major overlap of cytokinesis is what? Telophase. All right? So that's the stuff. Sorry, I'm very fast, but I have to be, because from the experience I have with the other class, I overstayed, and I don't want to do that with you, so that nobody will go away, especially when the most important thing comes around. All right? Okay. So this is just explanation of those things. So this is prophase. You see, everything is shortening and arranging themselves. Prophase. You see, metaphase, everything comes to the middle. That's metaphase, everything comes to the middle, M for M. Then we have division. The division starts at anaphase. Then they go to the extreme pose at telophase. And here Cytokinesis, the cell divides completely. Okay. Let me see if there is something. Oh, yeah, we have molecular timers. You need to know cyclins, you need to know CDKs. CDKs are cyclin-dependent kinases, all right? They combine themselves together to form what? Cyclin-dependent kinases complex. Extremely important that you know. Why? Because they control the DNA replication. That replication is controlled by what? Cyclin CDK complexes. They will ask you in exam. Replication specifically, all right? Aha. So these are everything that happens there. Now we have checkpoints. Checkpoint at G1, checkpoint at G2M, and checkpoint at the transition from metaphase towards anaphase. That's the third checkpoint. Okay, I'm jumping, I'm flying. We have the heredity, we have karyotype. You must know what karyotype is. It's a chart of this 46 chromosome. How many chromosomes do you have? 46. How many pairs of chromosomes do you have? 23. Hereditary is when the genetic information is transferred, transmission of genetic characteristics from one parent to offsprings. You see how handsome I am? Look at me very well. How handsome I am is genetic transfer from my parents, all right? Aha, specifically my mother. Thank you very much. Okay, so karyotype is 46 chromosomes laid out in a chart. Don't sleep. I can smell somebody sleeping. Don't sleep. So we have one pair of sex chromosome, all right? That's the X and the Y. Then we have the 22 pairs of autosomes, that's body cells. This is karyotype. This is karyotype. When you look at it, this is karyotype, all right? This is the 22 pairs that everybody have. Then this is, if you are a biological female here, you have this extra, that's 23 pairs. If you are a biological male, you have this extra, that's XY, all right? Now we have diploid, we have haploid. You need to know the differences between these two. When you talk about diploid, that's 2N. You are describing any cell with 23 pairs of chromosomes. That is somatic cell. That is body cell. If I cut myself here, if I cut myself here, the cell I will get from here is what? 46 chromosomes. Complete 46. That's 23 pairs. But if I take my sperm, that is sex cell. If I bring my sperm now, I'll be seeing only what? 23 chromosomes. Not 23 pairs, 23. That 23 chromosomes are what we call what? Haploid. They are usually sex chromosomes. Haploid. They are unpaired 23. It's usually found in sperm and in egg. But body cells, that's somatic cells, we call it what? Diploid. All right? Thank you so much. Diploid for dating. Thank you. Double. All right? Okay, thank you so much. So we have locus, that's the location of a particular gene on a chromosome. We have alleles, that are different forms of gene at the same locus. Then we have dominant allele. Some can, be very dominant, you understand, and some are recessive. Yes, you understand with questions, all right? We have phenotype. Phenotype and genotype. Genotype is alleles on individual positions for a particular trait. You understand? You can be homozygous if they are the same thing, identical. You can be heterozygous if they are different. Then we have phenotype. Phenotype is what you can see. You see how handsome I am? That is phenotypic appearance. All right? Okay. Then we have the Punnett square. It's just diagram where we cross match. All right. Let's go. Let me see. But then you need to know gene pool, please. Gene pool is the genetic makeup of whole population. You understand? And codominance is when two alleles are equally dominant. Incomplete dominance is when they are not, like when heterozygous individual shows phenotype intermediate between traits, like they are at the middle. You will see a question on that. You will understand more. Then we have the polygenic inheritance and pleiotropy. Polygenic inheritance and pleiotropy. I'll just use a question to describe it. Look at it. This is polygenic inheritance. What do you observe? You see gene 1, gene 2, gene 3. They join together to form what? Phenotype. One phenotype. You see? This is your eye color or my skin color. You see my skin color? It's not one gene that made it happen. It's several genes. So when several genes make one thing happen, we call it what? Polygenic what? Inheritance. All right? That's polygenic inheritance. So then this is pleiotropy. Pleiotropy is, when one gene is causing more than one effect. All right. For example, this gene for alkaptonuria, you will discuss it when you get to your clinical. It's causing this, just one gene is causing darkened ear, darkened sclera, and dark urine. Extreme, this is the normal urine, this is dark urine when standing. So when they describe a question like this, you should be able to know which one is polygenic inheritance. Genes are two or more loci contribute to a single phenotypic trait. Pleiotropy, one gene produces multiple what? Phenotypic what? Effects. Extremely important that you know this, all right? Penetrance, yes. Penetrance, percentage of population that shows a particular phenotype. Also important that you know this. What else? Okay, these are tumors. Tumors are just fast-growing cells. They can metastasize into the blood. You see how they are going. All right, somebody has a lung cancer, and the lung cancer is going to everywhere in the body. All right. All right. Now, carcinomas are very important, but I discuss it with you if I'm going to teach you pathology. I don't know, but carcinomas are for pathology, not for physiology. Remember, physiology is what? Normal. You see how I'm smiling? That is me, normal. If I frown, you'll be like, professor, what's the problem? Because you know me that I'm always smiling. All right. So normal is what? Physiology. Me always smiling is physiology. If I am not smiling, then you know that there's a problem. Oh, professor, you're not smiling. What's the problem? It becomes what? Pathology. All right. So physiology is what? Normal function. Function. Anatomy is what? Structure, or what? Location, what? Location. Extremely important that you know this. So when you are talking about pathology in terms of cancer, remember cancer is multiplication, all right? Or getting big. That's what cancer is. Rapidly, too fast, all right? So, there's something called tumor angiogenesis. That's when blood cells start growing in the tumor and makes the tumor to feed a lot on the blood. That's why a lot of, if you see somebody comes to the clinic and the person suddenly have, maybe if you're answering your NCLEX questions, for example, and they are giving you clinicals and, they ask you a question that somebody comes to the clinic, initially the person comes and the blood was around maybe 14G, and the person now comes down, the blood has decreased to maybe like 6 all of a sudden for no reason, and the person is having unintentional weight loss. You are thinking of cancer. Why? Because those cancer cells, when they are multiplying, they are taking the blood from the person. They are using that blood to feed and multiply. That's why they are always anemic. That's really important that you know this, right? And cancers are named for tissue of origin. Carcinomas, you are talking about what? Epithelial cells. Carcinomas. Lymphomas, lymph cells. Melanomas, pigments of the epidermis, all right? That's melanomas, all right? From melanin, melanocytes. When we discuss skin, you understand this, what I'm talking about. Then we have leukemia, that's in the blood. Then we have sarcomas in the bone. Terrible, all right? So that is cancer. Then these are the things that causes cancer: radiation, chemicals, viruses, and all whatnot. We have the tumor suppressor gene, because normally when something is growing rapidly in your body will suppress it. You understand? We stop it from growing. All right? That's what we call tumor suppressor genes. All right? Usually these cancers come and they destabilize that tumor suppressor gene. So nothing will suppress them when they're growing. They do a lot of things. They have a lot of effects. Replace functional tissues in vital organs, steal nutrients from the rest of the body, all right? Making the person there's something called cachexia. Sorry, my pronunciation is terrible, all right? It's severe wasting away of depleted tissues. Then they weaken the person's immunity and open door for opportunistic infection, just the way HIV works. This is Wilms tumor, but I'm not discussing Wilms tumor. For a physiology exam, you hardly get pathology questions. If you have a question, you can drop it in the box. And if you don't have, I'm jumping to my own questions. All right. Remember the rules and regulation. I'm going to stop projecting right now so that this thing can flow very well. Because if I'm projecting and putting in questions, it will be lagging. I'm even seeing that my mouth is okay. So we have 34 people here. I'm expecting 20 questions, 20 people to answer at least. If you don't know the answer, please put 911, emergency. If you want me to explain, I can explain. That's fine. All right. Yeah, so let's go there. Let's go there. Is there only 7 people that did like this. Please, everybody should do like let's get to 40. I'm so vain. Hey, God. Okay, I guess maybe you don't like it. Okay, let me see. Yes, keep liking, keep liking. Once we finish genetics, we jump to tissues. All right. Keep liking. That's 11. I have 40 people here. Keep liking and keep liking. Continue liking. Okay. Okay. I want to bamboozle you guys now. Bamboozle. But don't get too bamboozled. All right. So testing the microphone. Testing the microphone. Let me test the microphone with this question. Everybody must answer, please. If you don't know the answer, I'll put code blue or 911 or emergency. First question. DNA is a polymer of dash, which consists of a sugar, a phosphate group, and a dash. Answer. Answer. Let me give you options. Okay, fine. A, please listen so that I don't need to read it a second time. I'm trying to be fast because the other class, I took too much time. It's supposed to be 2 answers. Everybody's supposed to give two answers, all right? So A, amino acid nucleotide. B, nucleic acid nucleotide. C, nucleotide nitrogenous base. D, nucleotide nucleic acid. E, proteins nucleic acid. Come on, guys. Let's have it. Let's have it. Oh, wow. I started question and people ran away. It was 40 people before, and now they dropped to 22. What happened? Are they afraid of questions? Come on, guys. Let's go. Let's go. Let's go. Somebody says C. C is nucleotide and nitrogenous base. Okay, keep going. Keep going. Keep going. Yes, they ran away. Okay, they are coming back. It's not 24 people. Okay, you guys are correct. This is testing the microphone, all right? Testing the microphone. This is just a question to, ginger everybody. I need everybody to answer with me. If you don't know it, put 911. DNA is a polymer of what? Nucleotide, which consists of what? A sugar, a phosphate group, and what? Nitrogenous base. Nitrogenous bases can be divided into 2, which is what? The purines and the pyrimidines. The purine, they have double rings, and the pyrimidines have 1 ring. You should be able to know this. Okay. Don't worry, I'm still testing the microphone, testing the ground before I start firing in all cylinders. Okay. So I want to drop this question. I don't want you to answer until I finish the options, all right? I'll put the options in the question because it's an arrangement question. Which of these is the correct sequence from simple to complex? From simple to complex. Oh, somebody is still answering the other question. No, it's not this. C. C is nucleotide and nitrogenous base, please. So this question, which of these is the correct sequence? This is A. Hold on, let me put everything before you start firing me. It's A. This is... Let me see how hot you guys are. This is B. Incoming missile. Incoming missile. This is C. This is D. And this is E. Somebody's already firing. Okay, so that's the options. Which of these is the correct sequence from simple to more complex? Guys, come on, hit. Come on. Give me the answers. Give me the answers. Keep going. Make sure you don't follow anybody when you're answering. Make sure you read the question very well, understand it, and answer. Somebody says carbon, nitrogenous base, nucleotide, DNA, chromatin. That is E. So that person choose option E. Okay, keep going. Can I repeat the question? The question says, which of these is the correct sequence from simple to more complex? From simple to more complex. I put the 5 options, all right? Okay, many people are saying B. Somebody says E. Somebody says E. So keep going, keep going. I need somebody say E and B are the same. E and B are not the same. E and B are not the same. Let me see what I posted there. Is E and is. Oh, hold on. No, it's not the same thing now. No, it's not the same thing. I was shocked. It's not the same thing. Keep going, keep going, keep going, keep going, keep going. Okay, so the answer is E, actually, not B. Not B. The answer is E. Think about it. Everything starts from carbon. Carbon forms the nitrogenous basis, right? Then the nitrogenous basis forms what? Nucleotide, as we mentioned. What does nucleotide form? Nucleotide form DNA. When you now tie the DNA with protein, it becomes what? Chromatin. Remember, chromatin must be the tied one. All right. B says carbon, nitrogenous base, nucleotide. No, you don't have chromatin before DNA. You have DNA before chromatin, because chromatin is when you tie the DNA with protein. It's more complex. It's more complex. You tie it with histone. It's more complex. So please, I need you to get it right. All right. Nucleotide, DNA, chromatin. The answer is E. I move. If you are confused, you can just tell me confused and I will come back. But if not, I need you to answer this question for me ASAP. I need us to be faster, right? If I just put this question 1 minute, I need all the answers in 1 minute. We are 30, more than 30 here. I need all the answers in 1 minute. Somebody say answering. Okay. Look at this question. Guys, look at this question. A dash is composed of granular cluster of 8 proteins called dash with DNA 1 around them. A dash is composed of granular cluster of 8 proteins called dash with the DNA 1 around them. A, nucleosome, centrosomes. B, nucleosomes, histosomes. C, chromosomes, histones. D, chromosomes, centromere. And E, centromere, chromatin. Quickly. Quickly, guys. Quickly. Okay, we have B, we have E, nucleosomes and histones. Keep going. Keep going. Somebody is texting me. Keep going. Okay, yes, the answer is what? Nucleosome and histones. The answer is B, guys. Please bear that in mind. I'm jumping. Perfect. This is the question that I have been looking for. This is the question you can never escape. You cannot escape this. And I'm not giving you options. Please, I'm not giving you options. Oh yeah, let's go. Let's fire. I need us to finish on time. We already spent 44 minutes. Jesus. The time runs so fast. What is the complementary DNA base sequence to the DNA strand TGCCAT? Answer quickly, guys. Somebody's asking me question mark. It's okay. Just put your answer. We will see to it. All right. It's okay. No question, no answer is wrong, except when I say it's wrong. Keep going. Less than 1 minute for each question. Keep going, guys. Okay. Yes. The answer, look at it. T is A, right? So you write A. Let me write it. Let me see. Well, what do I have? I have T, G, C, C, A, T. So T goes with A. G goes with C. C goes with G. Another C goes with G. Then A goes with T. And T goes with A. So the answer is A, C, G, G, T, A. That's the answer. A, C, G, G, T, A. If that's what you write, you are correct. You are very, very, very correct. Okay, so let me move. Yes, somebody is giving us mnemonics. Perfect. Thank you so much. You've been so helpful. Apple tree, car, garage. Okay. Apple tree, that's A and T. Okay. All right. So give me the answer to this question extremely fast, guys. Just the way you've been fast. Let's finish this quick. Answer. A dash contains the necessary information for the production of a molecule of RNA. A what? A what? Do you want options? Keep going. Keep going. Keep going. Yes, somebody say ACGGTA. Yes, I think you're correct. ACGGTA, yes. Okay, keep going, guys. Somebody say genome, somebody say gene. Keep going. Let me get 5 more people to give me the answer. Dash contains the necessary information for the production of a molecule of RNA. Thank you so much. The answer is not genome. The answer is what? Gene. Genome is the entire genes, an individual have. The answer is gene. Please. The answer is what? Gene. So all the sets of DNA in somebody is known as what? Genome. All right? All of the DNA in a set of 23 chromosomes is known as what? Genome. So you need to differentiate genome and what? Gene. All right? And remember, I told you that the human genome is 3 billion. There's a question about it, but I don't want to waste my time with that question because I know all of you will get it. Okay, look at this question, guys. Look at this question, guys. Copying genetic information from DNA into RNA is called dash. Copying genetic information from DNA to RNA is called dash. Using the information contained in mRNA to make a polypeptide is called dash. Put in the answers, guys. There has to be 2 answers. So somebody is writing only transcription. There has to be 2 answers. Option A, transcription, translation. Option B, translation, transcription. Option C, DNA replication and translation. Option D, DNA duplication. DNA duplication and transcription. Option E, DNA translation and RNA transcription. Okay, you guys are so correct. Somebody said translation. You guys are so correct. The answer is what? Transcription and what? Translation. Copying genetic information from DNA to RNA is transcription. Using the information contained in this thing to create protein is called a polypeptide. Is called what? Translation. All right. You see how I'm saying transcription, translation. I know you'll be laughing there. Don't laugh at me. Okay. Let me put this question now. If you fail it, I will laugh at you. You that is laughing. I can see you. Which of the following is not directly involved in translation? Options. She's laughing. Option DNA, mRNA, tRNA, rRNA, ribosome. Answer. Fire, fire, fire. Fire. It seems this class is really, you guys are really hot too. Yay. Okay, because I'm giving you questions. Okay, this kind of question. Okay, wait. Okay, wait. Say all of you are answering DNA now, very quick. That's good. Okay. We are still testing the microphone. I don't think this is the main question. We are testing the microphone. Yeah, the class is hot. It's too hot. Like I'm feeling the heat. I'm feeling the heat. Okay. Look at this question. Let me see how hot you guys are. Let me see how hot you guys are. Oh, somebody said ribosome. I don't know why you said ribosome. Is ribosome in the option? No, the answer is not ribosome. Oh. Ribosome is involved in translation. Remember, the translation itself happened in ribosome. That's the kitchen. All right. mRNA is part of the translation because it brings the code, the information. tRNA, remember, it brings the groceries, which is the amino acids. And rRNA is the chief chef. All right. So all of them are involved, but DNA is happening in where the nucleus. It has nothing to do with translation. Instead, it has everything to do with replication and transcription. Okay. I believe you understand that. Somebody said, I am this. Okay. Does it mean I'm fine? Okay. I believe you are saying you are fine. Transcription occurs in the, let me see, nucleus. Okay. You guys are threatening me with your answers. You are getting it too correct. But most translation occurs in the cytoplasm. All right. Perfect. You see this question? It's one of the most important questions you can meet in genetics. Look at this question, guys. The dash are turned on or off by regulatory proteins in accordance with changing needs for the dash they encode. Answer. Options: A, enzymes products. Option B, ribosome proteins. Option C, gene proteins. Option D, proteins gene. Option E, genes ribosome. They cut me. You guys cut me. You guys cut me. Okay, fine. Okay, so the answer is genes. Genes are turned on or off by regulatory proteins in accordance with the changing needs for the proteins that they want to encode. All right. When they want to make long hair, you cut your hair. You want to make long hair, it turns it on. When your hair grows too much, it doesn't want it to overgrow, it turns it off. All right. That's regulatory proteins. Okay, let's see this. I am sure some people will get this question incorrect. After translation, a protein may undergo structural changes called dash modification. What's the answer? What's the answer? The answer options: splicing, post-transcriptional modification is B, post-translational modification is C, polyribosomal is D, and secretory is E. Let me see. Am I saying post-transcriptional? Okay, it's post-translational. I'm dramatic, I know. Fine. I know that's what you're saying in your mind. You're saying this professor is too dramatic. Fine, let it be. Okay, yeah, you guys are correct. It's post-translational modification. Oh, okay. Let me see if you get this. I know you're right. I said this guy is too. Okay, look at this question. Please don't answer. Let me pose the options, all right? This is the question. Since there are no genes coding for carbohydrate, nucleic acid, and what? And nucleic acid or lipids, how do cells produce them? This is the option. So you pick the options. This is A. This is B. Incoming. This is C. This is D. Okay. No, I'm not giving you the last one. Please, my hand is paining me. This is A to D. Give me the answers. Come on, guys. Come on. Come on. Move it. Move it. Move it. I think the first one. Stop thinking. Okay, so I'll take it that you picked A. Since there are no genes coding for carbohydrate, nucleic acids, or lipids, how do cells produce them? A, enzymes produced encoded by genes synthesize this product. Is that the answer? Yes, you guys are correct. Enzymes encoded by genes synthesize these products. All right. So the gene cannot make them. There's no gene for them. So those genes make enzymes. The enzymes now synthesize them. Thank you so much. You guys are the best. You guys are the best. The answer is A. So if you see something similar in the exam, I trust you will be able to get it. Let's see. Oh. You guys are feeling each other, right? You are feeling yourself. You are feeling yourself. Okay. Let me see if you can get this one. You are feeling yourself. I know in your mind you say, this thing is very easy. Yes, it's very easy. But fine, let me see if this one is easy for you. Now I'll give you the options. Hold on. Let me give you the options. Hold on. This is A. Hold on. This is A. Okay. This is B. This is C. This is D. And this is E. Okay, let's go. Let's go, guys. You were able to radioactively tag and thereby trace an amino acid that is used to make insulin, a hormone that will be exported out of the cell. The pathway of the tagged amino acid would be dash. What is the pathway? Come on, guys. Come on, we're up to 40 here. Come on. Push it. Push it. Punch in that answer. If you are not sure, just write and not sure. All right? All right, 911. Let's come and rescue you. Not sure. Okay. Good. Somebody said, let me see. Complex Golgi. Okay. Golgi complex Golgi. Just pick the particular option you are talking about because like I'm finding it difficult to see what you wrote. Oh, somebody initially says rough Golgi extra. Okay, not sure. Okay, guys, no smooth. Not sure. Okay, guys, the answer is B. The answer is B. Rough ER. From rough ER where it's made, Golgi complex is packaged in Golgi vesicles, those Amazon boxes. Remember I told you shaping and reshaping. If you watch a prior video on genetics, it's talking about what? Amazon. You understand? Packaging is done in Amazon. So production in rough, packaging in Amazon, that's Golgi complex. It packages it into vesicles in boxes. Vesicles are boxes, and it pushes it out of the cell. That's into the extracellular fluid. The answer is B, guys. That's how you need to think about it. Oh, I love this question. I love this question. See this question, guys. Let me see who gets this question. I'm not giving you option. Let me see who gets this question. Option 20, 51, 102, 153. I've not even given option. No. These people are wonderful. Yeah. Who did not understand how we got the answer? The answer is 153. You guys are wonderful. Yes, keep liking, keep clicking like, guys. Keep clicking like if you are enjoying this session. So it's 51 times 3, yeah, because we base triplets, all right? Yeah, that's how 51 times 3 will get 153. That's how we got it. You guys are the best. Let me see this question. This question is too easy. Okay. You guys are hot, right? Okay, so let me try you with this question. I need to see a question that will conquer all of you. You guys seem to be conquering all my questions. What is happening? Don't I have stronger questions for you? DNA polymerase is most active during which phase of cell cycle? Options. Let me give you the options. Options S, G1, G2, M, GO. This is sweet, though. Yes, it's S phase. Yay. Yeah, you're answering this question like without even consideration. No joy, joyless. You guys are so hot, man. Okay, so for people that don't understand what this question is about, this question is asking about DNA polymerase. What does DNA polymerase do? You say chicken. DNA polymerase, you guys are so happy. DNA polymerase replicates, correct? It causes what? Replication. All right. And where does replication happen in cell cycle? That's the S phase. You guys are wonderful. Okay. I was thinking I'm going to tempt you guys with that question. If you know that one, then I'm not asking you this next one. This next one says the G1, S, and G2 phases of the cell cycle are collectively called what? Type in the answer. I'm not putting the question, guys. G1, S, and G2. Collectively, what are they called? I don't want to put in the answer. The question. I don't fully participate in DNA replication except. Okay. The answer is interphase. Yes, you guys are good. I've confirmed it. But I'm not giving up in conquering you guys. I've not given up. All right. So look at this question. I'll call the options by mouth. All right. Yes, it's interphase. You are good. All of the following participate in DNA replication except option A, DNA, option B, ribosome, option C, DNA ligase, option D, DNA helicase, option E, DNA polymerase. A, DNA, B, ribosome, C, ligase, D, helicase, E, polymerase. Answer. Which of the following participates in DNA replication? You guys are perfect. Except. So all of them participate apart from ribosomes, all right? Oh, look at this question. Yeah, you guys are on fire. You guys, don't burn me up, please. I'm the only son of my parents. Don't burn me with fire, all right? Cytokinesis overlaps with which phase of mitosis? Somebody said interphase. Somebody said telophase. Somebody said tel. That's telophase. I get it. Keep going. Somebody said telophase. Keep going. Somebody said... The rate people are getting these answers. I think we are going to finish this very fast, though. I love it. I love it. Okay, look at this question. Telophase. I played too much. Look at this question. Dash opens up one segment of the DNA helix during replication. Dash adds complementary free nucleotides to the exposed DNA strands. Answer. I'm not giving option. No. You guys are so hot, so I'm no longer giving you options. You have to tell me the answer, and I will tell you if you're correct or not. There are two answers you have to put because it's dash and dash, right? There are two answers. So I want you to write the two answers together. If you write one, we'll start looking for one in another country. You understand? In Mexico. You see? Someone has written one. By the time the person is writing the second one, now we'll see it in Japan. Please write the two of them together. Is I should give option? Oh, somebody has given up. I love it. Answer. Option A, DNA ligase and DNA helicase. Option B, DNA helicase and DNA polymerase. Option C, DNA ligase and DNA polymerase. Option D, DNA helicase and DNA ligase. Option E, DNA polymerase and DNA ligase. What I'm just hearing is DNA. Someone say option C. Option C is DNA ligase and DNA polymerase. Okay. So you guys are doing well. Keep liking. I'm seeing 17 like and I'm seeing 40 people here. Why? You don't like me? Please keep liking. Click that like button. Okay, so the answer is a B, option B. You're correct. All of you are correct. DNA helicase and DNA what? Polymerase. You see the question, what opens up? That's unzipping. That's helicase. It's unzipping. And that adds complementary free nucleotides to the exposed DNA. That's replication, creating the other arm of the DNA. That's DNA was polymerase. All right. Okay. Okay. You see this question? This question, I'm expecting everyone here. Yes, keep liking, keep liking, keep liking. So is your like button spoiled? I don't know. I don't understand why we have 40 people here. We have 17 likes. Okay, maybe there is love that they are giving me. That's why it's not reflecting on the like. I get what you are doing. Okay. See how the love is going up. This thing is affecting me. It's getting me emotional. All right. The stage at which chromosome aggregate along the equator of a cell is dash. Where do they aggregate along the equator of a cell? Where? Keep going, keep going, keep going, keep typing, keep typing, keep typing. Yes, equator is middle. I agree. Keep typing. Yes, M for M. M for M. The answer is metaphase. M for M. M for M. Yes, you're correct. Okay, you guys, I've not seen any question that is overcome you guys. So and I'm almost done with genetics. Ha. No, I don't know if it's a good thing or a bad thing. Is it that I cannot come up with a strong question? Sister chromatids are pulled to opposite poles of a cell during dash of the cell cycle. Okay. Option S phase, B, G2 phase, C, prophase, D, metaphase, E, anaphase. Logged out? Are you guys being logged out? I don't think so. Because I'm monitoring the number of people here. You guys have been just opposing between 30 and 40. So probably some few people is logging out, some few people, or some few people are going out. A, S phase B, G2 phase C, prophase D, metaphase E, anaphase. You guys are correct. Ana away. Someone say Ana away. Okay. That's anaphase. Okay. This person likes a whole lot of mnemonics. I love it for you. I love it for you. Okay. Look at this. Look at this question. I keep having to log in. Wow. Why? Are you using your phone or you're using your laptop? I think you should use your laptop. It will help more. Chromatin shortens and thickens, coiling into compact rods during dash of mitosis. Oh, laptop? Maybe you should use your phone and see if it's better. But what you need to know is that why I'm using YouTube is that at the end of the day, it will save it on my YouTube stream channel, on my channel, right? So you can always just come to the channel and look at what you miss. All right. Chromatin shortens, thickens, coiling into compact rods during dash of mitosis. Telophase, anaphase, interphase, prophase, metaphase. You guys are correct again. Why? This is prophase. Yes, you're correct. Why are you people too correct? Yeah, okay. You see how I smile? That's called evil. In Nigeria, it's called evil smile. It means that I'm bringing a banger. I'm bringing something that will shake you. Okay. Okay. Somebody is laughing at me or laughing with me. All right. But let's see. Let's see. Look at this question. I'll give you the option. It's okay. It's okay. It's okay. I'll give you the option. Which of the following omits some stages of the cell cycle? Calm down. Let me give you the option. Now tell me which one omitted a step. This is A. I am sure that this question will shake you guys. There's nothing you can tell me. There's nothing anybody can tell me. This is B. This is C. This is E, D rather. And this is E. Mind you, they didn't say which one is the main sequence. They say which one omits. Which of the following omits some stages of cell cycle? Let me see. Oh, D. Oh, these people caught me again. Ah, I'm done with you guys. Okay, so for people that don't understand it, if you look at it, G1, S, and G2, all right, are interphase. Then the next phase is mitosis. So it's correct. Second one is what? Interphase and mitosis, correct. Third one is G1, DNA replication, that's S, G2, and mitosis, correct. The fifth one, that's E, says interphase, prophase, metaphase, anaphase, telophase, all of them, and cytokinesis. So interphase and mitosis, correct. But the answer is D, which is prophase, metaphase, anaphase, telophase, cytokinesis. What about interphase? It's not there. All right, that's what is missing. So that's why D is the answer. So always look at the question properly before you pick, please. Don't just go and pick. Okay, look at this one. A karyotype shows the dash chromosome sorted and isolated from a cell in the dash. Options: 23 interphase, B 23 metaphase, C 46 prophase, D 46 metaphase, and E 46 S phase. Twenty-three interphase, 23 metaphase, 46 prophase. That's why I don't like posting these options like this. It will be a little confusing. 23 interphase, 23 metaphase, 46 prophase, 46 metaphase, and 46 S phase. Answer. Keep going. Keep going. Are you guys now afraid of? You see, I told you I would shake you guys. You guys are now afraid of this question. Is there anybody that's still yet to answer? Okay. Nobody. Okay, you guys are so correct. It's 46 metaphase. 46 metaphase. I like the fact that everybody's understanding so that I don't need to keep explaining. It makes make sense to me. Now, I will shake you guys. There's nothing anybody can tell me. Look at this. Tell me the answer. Ha ha. Ha ha ha. Ha ha ha. According to that metaphase, yes, correct. Okay, tell me this answer. A cell finishing mitosis has dash DNA molecules. Why cell finishing DNA replication has dash DNA molecules? Let me see. Okay. You guys caught me again. It's 4692. All right. A self-finishing mitosis has 46 DNA molecules. Now, when DNA replicates, it becomes times 2. That's 92. All right. Yes. So that's how the answer is 4692. 4692. Yeah, I should have asked you this question, but this recessive and dominant, you will get this answer. No. No. Even this one, Seth, I don't know if I should be asking this question because I am sure that with the kind of hotness you guys have, you guys will answer this one. Oh, yeah. See, this I should ask. Eye color is caused by genes at multiple loci. The phenomenon is called what? This one, everybody will get this one. I don't like this question. It's too cheap. It's too cheap. It's too cheap. It's too cheap, but a lot of people are not typing. You know. We have 35 people here. We have 4 people. That means it's difficult too. I should be happy. It's difficult. Oh, a lot of people have answered finally. It's polygenic inheritance. Fine. 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 I'm angry. You guys need to be failing some of these questions so that I'll be like, okay, I'm teaching something. You're getting everything. I feel useless. He says too cheap. Okay. Oh yeah, look at this one now. Fine. Oh yeah, answer this question. I'll give you the options. A, 23 somatic. B, 23 haploid. C, 23 diploid. 46 somatic and 46 diploid. They're getting it again. Yeah, somebody has to fill it. Let me see. Somebody will fill it. Fill it. Hi. People have sworn that they have conquered genetics. Okay, 23 haploid. Okay, you guys are. Please, I'm now depressed. Thought I was bringing hard questions, and don't know it's you guys are. It's now cheap, easy questions for you. All right, answer this one. If you think you know this question, answer it very fast. Let me see. Very fast. Don't think when you're answering it. Very fast. Let me see. Why some people are texting me that they didn't get the link? I thought I sent the link to everybody. Why are they saying they don't give? Okay, yes, the answer is somatic cells diploid. So somatic cells are diploid, sex cells are haploid. Okay, I'm not asking this question again. It's too cheap. Please, let me jump to something that's more serious. Genes produce their products. Okay, cells undergo divisions when which of the following conditions have met. No, this one, you people get it. Oh, let me ask the last question for today, because I forgot that we have tissues and this thing. Let me ask you the last questions. The last question for today. I know it's a giveaway and it's fine, no problem. I'm crying. Last question for this. Which of the following is not found in the RNA? I like this session because we are kind of faster. By this time, last 2 days ago, we have not even gone half. Which of the following is not found in RNA? We have thymine, we have uracil, we have ribose, we have guanine. This is my last question. Just fail it, please. Get it wrong. People are so stubborn. Okay, you guys are so correct. The answer is what thiamine. Remember, thiamine in DNA, uracil in RNA. All right. Our option is thiamine, uracil, ribose, and guanine. So in RNA, we don't have thiamine. Okay. Thank you guys so much for this. So let's jump to tissues, please. Let's jump to tissues. I'm gonna just use 10 minutes, please. 10 minutes to rush that through tissues. Then I bombard you with questions. Why I always use 10 to 15 minutes to channel those things. But for skin, I'm not going to open it. I'll just give you questions for skin. You understand? So skin will be more direct question because I know by the time we are getting to skin, a lot of people here will be tired. I'm sure of that. So I won't want to hold them for too long. So, but tissues, I'm sharing my screen now. You guys are the best. Sharing my screens. Don't sleep, oh. Don't sleep. Don't sleep. We have other things. Don't sleep. Okay. If you can see my screen, please click. Tell me. Just say you can see it, please. Tissues. Can you see my screen? Just say yes. Okay, yes. Somebody say yes. Somebody say we can't fail. I am sure you can't. I am sure. With the way you guys have destroyed my questions this night, I give up. I give up. Now, if they want to, if they are looking for people that will go do a competition in genetics, you know, you guys can volunteer. All right. So let's jump into tissues. We don't have much time. I really want to leave you guys at least in 2 hours, 30 minutes, 1 hour, 30 minutes more. We already spent, let me see how many minutes we spent. We've already spent 1 hour, 23 minutes. Wow. And it's looking as if we've not even started. Like everybody is so energetic. And I love that for you guys. I know some people need to sleep because our class is, for people that have class with me tomorrow, it's early. So I'll be fast with you guys so that I can go crash. Okay, so when you are talking about tissue, the 4 broad categories of tissue are epithelial tissues, connective tissues, nervous tissues, and muscular tissues. You know how I fly. So just don't write. I don't need you to write. All you can do for me is just to pay attention, all right? If you are not seeing my screen, if I'm lagging too much, just tell me. You understand? I will do something about it. So there are 50 trillion cells of about 200 variety, all right? 4 broad categories. We have the epithelial cells, connective tissues, nervous tissues, muscular tissues, all right? These make up what? Organs. Extremely important for you to know. Now, when you are defining tissue, you are defining the types and the function, the characteristics of the matrix, the relative space, all right? Relative space of each and every one of them. And you need to know about clear gel, you know, because we have fibrous proteins and we have clear gel ground substances. Among that clear gel ground substances, we have tissue fluid, we have ESF, we have interstitial fluid. Please, I don't want you to mistake interstitial fluid with intracellular fluid. Intracellular fluid is inside cells. But here we are not talking about cells. We are talking about what? Tissues. Cells make up what? Tissues. So because there is cell here, the fluid outside them is called extracellular fluid. So all tissue fluids are what? Extracellular fluids. That's what I need you to know. All tissue fluids are what? Extracellular fluids. All right? Sorry. Now, remember ectoderm, endoderm, and mesoderm. Ect is outside the skin, epidermis, and what? When you touch me, I can feel it. That's nervous system. That's ectoderm. That's how you remember it. Then when you're talking about endoderm, what are you talking about? Endoderm gives rise to what? Things that are inside. In. That's the digestive system. All right? So if I throw something down here, it comes out from my anus. The only way it cannot come out from my anus is if it enters into the middle, which is the mesoderm. And what are the mesoderms? Derivatives. We have cartilage, we have bone, we have blood. CBB or BCB, BBC, BBC. Bone, blood, cartilage is mesoderm. Epidermis, nervous system is ectoderm. Every other thing is what? Endoderm. If you remember it a lot for this level, you are good to go. All right? Just make life easy for yourself. Don't overstress. Remember, the histologists, they prepare and preserve slides and section tissues. Very important. Smearing, spreading, also important part of sectioning. All right. Now, this is where they usually ask questions: longitudinal section, cross section, and oblique section. They will ask you, tissues that are cut on its long axis is called what? Longitudinal. Tissues that cut perpendicular is called what? Cross. Tissues that cut at angle between cross and longitudinal sections is known as what? Oblique. Extremely important for you to know this. All right. Now, when you're talking about epithelial tissues, we are taking them one after the other. Remember, how many do we have? We have four principles: epithelial tissues, connective tissue, nervous tissues, and muscular tissues. This is a crash program. All right. If you've not subscribed, please go and click subscribe, because other videos that are going to come up, you have to see them. All right. Now, epithelial tissues, that's what we are discussing. After that, we'll go to connective. So you have to know the functions of epithelial tissues. I'm not going to talk about it. There's basement membrane, where the epithelium is what? Standing upon. And basement membrane separates the epithelium from what? The underlying connective tissue. I told you that epithelium is what? Avascular. It does not have blood vessels in it. So how does it feed? It's a vampire. It's feeding from the connective tissue. Which of the connective tissue? The loose connective tissues. They are the ones that are most vascularized. So remember the basal surface and the apical surface. Now, this is a very interesting page, all right, picture. You understand? This is classes of epithelium. You understand? You can classify epithelium based on their classes and based on their cell shapes, based on the layers and based on the cell shapes. Yes, areola, which is type of loose connective tissue, based on the layers and also the cell shapes. In terms of layers, we have simple, we have pseudostratified, and we have what? Stratified. Simple, pseudostratified, and stratified. Simple is one layer. Stratified is anything more than one layer. If there are some that are not touching the basement membrane, they become stratified. If you look at this third picture by your left, upper left, this place I'm pointing, you see all of them standing on each other. All right, and it's flat. But the most important thing is that they are stratified. Here is simple. Now, this middle one is pseudo-stratified, means that it's trying to confuse you that it is stratified. It is false stratification. It's not actually stratified, but it's trying to confuse you. And the only one that has pseudo-stratified is columnar. So we have pseudo-stratified what? Columnar. And we have simple columnar. All right? Okay, so this is simple columnar. This is pseudo-stratified columnar. This is stratified what? Squamous, because it's flat. Now, if you look down here, the flat cells are what? Squamous. The cube cells or the cup-like looking cells are what? Cuboidal. And we have these tortoise cells that are columnar. This is columnar. Even this part is what? Columnar as well. So you need to understand that these tall looking things are what? Columnar. This one that is the same shape, length and width, is what? Cubes. And this one that are flat is what? Squamous. All right. Now, the ones that are one layer, that are the simple, we have simple squamous, simple cuboidal, simple columnar, and pseudostratified columnar. All of them are one layer. Simple columnar are thin, and simple squamous are thin and scaly. Simple cuboidals are round. If they ask you which one is round, simple cuboidal. Round and square, simple cuboidal. But which one is tall and narrow? Simple columnar. You cannot forget this. First appearance of stratification, pseudostratified. And this pseudostratified and simple columnar, they usually have what? Goblet cells. And I told you that pseudostratified is found where? It is found where? In the lungs, right? Specifically around the trachea. That's where you see the pseudostratified. Why? Because it has goblet cells, and that goblet cells produces mucus that is trapping, that's climbing on top of the cilia, trapping the dust and all the stressful things that you take in. All right, it's trapping them. That's first line defense. All right, so the respiratory tract, you see it. But in the respiratory tract, you can also see simple squamous. All right, we have it in the alveoli. Don't forget, you have it in the alveoli, you have it in the kidney as well, specifically in the thin descending loop of Henle. That's where you have it. All right, please remember the goblet cells. Very, very important. Now, this simple squamous, remember it in the alveoli. You see, this alveoli is extremely important. That's where they will ask that question. It permits rapid diffusion and transport of substances. Simple squamous. All right. Now we have simple what? Cuboidal. This is simple cuboidal. Cup. Anywhere that they make fluid, you see it, like liver, thyroid, mammary gland, salivary glands, bronchioles, kidney tubules. That is where you find cuboidal. All of them make fluid. And where you do put fluid? Inside a cup. That's why I said remember it as a cup or as a cube. Cuboidal. All right. Now, let me use this opportunity to also talk about the stratified cuboidal. Stratified cuboidal, the fact that it's stratified is strength, is stronger. So it's found in the ducts where this fluid moves, like the seminiferous tubule, like ducts. All right, mammary ducts, all the ducts. That's where you see this stratified cuboidal. But the one that is producing the fluid itself is called simple cuboidal. Same thing with this guy here, simple squamous. Simple squamous is simple. It allows fluid to pass through it. But if we need it to be stronger, what do? We put it on layers, because what? Stratified squamous. So you see stratified squamous where is things usually pass through. Like where? Like the esophagus, the digestive tract, the anus, the vagina. All right, even the mouth. You see all of them stratified what? Squamous. Stratified squamous. Even the skin, you see stratified squamous. The only difference between the skin and inside the body is what? Keratinization. But there is a part of the body that is also that's inside that is keratinized, which is the hard palate up. So you need to know this and know peace. All right. So I jump. This is simple columnar. You usually find it where? In the GI. That's where they will ask you this simple columnar. GI and also the uterus and uterine tubes. GI, uterus and uterine tubes. Extremely important. Simple columnar. Simple columnar also has goblet cells and it has cilia, just like pseudostratified. Put that in your head. This is pseudostratified, all right? It's confusing you, trying to make you think that it is stratified, but it is not stratified. It also has goblet cells and it has what? Cilia. Now, the two major difference, the major way you can identify this, differentiate the pseudostratified and this columnar is that columnar, simple columnar, all of them are equal in the apical and in the basal surface. All of them is touching the basal surface and touching the apical surface. So if they ask you a question, which of these will you see all of them touching the apical surface and the basal surface, and they are single row of tall, narrowed cells? We are talking about simple columnar. But in pseudostratified, all of them are not touching the apical, and all of them are not touching the what? The basal. That's what you need to know to differentiate them. And where are they found? The respiratory tracts. Always very important for you to know. All right. So let's jump to stratified. Which one is very rare stratified? Stratified columnar. You can never hear stratified columnar. You can never hear it. All right. And which one is the most widespread? It is stratified squamous. So we have stratified squamous, we have stratified cuboidal, we have stratified columnar. Then another stratified is transitional epithelium. This transitional, we usually find it in the ureter and in the ureter and the bladder. Transitional. It changes shape. Transitional, ureter and bladder, but it is stratified. All right. So you have to remember this. Know it and know peace. Keratinization, skin. Usually they are dead surfaces. All right. Let me see. So this is keratinized stratified squamous epithelium. You find it on the skin. You see epidermis, palms, soles, and all those things. Please don't sleep off on me. Oh, I can smell somebody sleeping. We are tired. 4 people run away. You want to don't run away. Oh, the questions are coming. All right. This is non-keratinized. You find it inside the body, like tongue, oral mucosa, esophagus, vagina. Inside, even the anus, inside. All right. Very, very important for you to know. All right. Let me use this opportunity to talk about the vagina. The vagina, initially, when somebody is pre-puberty, the vagina is a simple cuboid, right? Now, but, oh, we are perfect. Thank you so much. But when you hit puberty, the vagina becomes stronger, changing from simple cuboidal to what? To stratified stratified squamous, non-keratinized. It has to become strong. That's why it's stratified itself. Becomes strong. Because when, during sex, the penis goes in and out of it. During childbirth, the head of the baby and the body of the baby comes through it. If it is not strong, it will not allow these things to pass through it. All right? So that's why it changed. So that change from simple cuboidal to stratified squamous, non-keratinized, is called what? Metaplasia. Meta what? Plasia. Meta what? Plasia. All right? That is that change. It's called physiological metaplasia. And smokers has what? In their lungs, according to the respiratory tract, we have pseudostratified, right? All of you know that we have pseudostratified. I know I'm talking too fast. I'm sorry. Later you watch this video and you slow it down. I'm sorry, but I need to save time for everyone. So in the lungs, you see, in the respiratory tract, you see what? Pseudostratified epithelium. For people that are chronic smokers, that pseudostratified will change and become what? And change and become what? Stratified squamous non-keratinized, just like the vagina. And remember, stratified squamous does not have what? Goblet cells, so it cannot produce mucus. So that first line of defense for your respiratory tract has been eliminated. So if a little dust enters, what will happen? The second line of defense comes into play, which is what? Coughing. That's why you see chronic smokers cough a lot. So if I pass through dust right now, I don't care. I won't even cough. Why? Because I don't smoke. I still have what? Pseudostratified epithelium producing mucus that is trapping all the dust particle. But somebody that's a chronic smoker, it has changed. So there's no mucus to trap. So what will happen? The person will start coughing. Mechanical reflex. All right? So that change from pseudostratified to stratified squamous is known as what? Metaplasia. Just like the vagina. But the vagina is physiological. This one is what? Pathological. It's a sickness, all right? Because most metaplasia, pathological metaplasias, most of them lead to cancer. Just like when you are having GERD, GERD, all right? What happens? You see acid reflux. And the stomach is acidic, but the esophagus is not. So if you keep burning the esophagus, before you know it, it changes the epithelium of the esophagus. That is metaplasia. And most times, metaplasia, if it continues for a very long time, it can. Science has proven that it can change to what? Neoplasia, just like the lungs. Because metaplasia, we beget what? Neoplasia in the lungs. All right? Okay. Now we have stratified cuboidal epithelium. I told you it is on the ducts, also on the seminiferous tubules. Remember that. We have transitional, which I told you is the ureter and the bladder. Remember that. Now, let's talk about connective tissue very fast. Connective tissue, they are very diverse, abundant type of tissue in which cells occupy less space than the matrix, all right? They help to support. They are highly vascularized. The most vascularized are the loose connective tissue, and the least vascularized are the cartilage. Remember that. The cartilage. These are the functions: supports, connect, immune protection, and all those things. Look it up. These are the cells: fibroblasts, macrophages, leukocytes, plasma, mast cells, adipocytes. They will tell you which of the following is not a cell. I will show you a question on that. Now, in terms of fiber, we have collagenous fiber. Collagenous fiber, example of collagenous fiber is tendons, ligaments. Tendons, ligaments, that's collagenous fiber. Very flexible, all right? Now, we have the reticular fibers. You understand? Example, where you see them is spleen, lymph nodes. Then we have the what? Elastic fibers. These reticular fibers can also stretch. That's why when they are inflamed, when lymph is inflamed, it becomes so swollen. Same thing with the spleen. Now we have elastic fibers. Elastic fibers, they stretch a lot, all right? They stretch a lot. And they contain elastin. Very, very important for you to know. Okay. Now we have ground substances. Example of ground substances, they are usually gelatinous. They usually have gelatinous to rubbery consistency. All right. Example, GAGs. That's glycosaminoglycans. All right. Glycosaminoglycans. What do? This thing is very important. They regulate water and electrolyte balance of tissues. This thing extremely important. They regulate water and electrolyte balance. Example. Further examples: we have chondroitin sulfate. We have heparin. We have hyaluronic acid. They will tell you which of the following, does not help in gelatinous substance consistency. You have the GAGs, we have the chondroitin, we have the heparin, we have the hyaluronic acid. We also have the proteoglycans, all right? Proteoglycans. So you need to remember this. Now, in terms of connective tissue, remember we have the connective tissue proper, and we have the specialized connective tissues. Now, the connective tissues proper, what do we have? We have the loose connective tissue and we have the dense connective tissue. The loose connective tissue, what do we have? We have the areolar, we have the reticular, we have the what? Adipose. The dense connective tissue, what do we have? We have the dense regular, we have the dense irregular. Extremely fast. Somebody sends me a message. Somebody said, Nchebe, you are talking so fast. How do you generate what we are saying? I'm sorry. I just want to finish up, all right? So let's look at it. So this is the diagram. This is areolar tissue. Areolar tissue, very, very, very disorganized, but it has a lot of blood tissues in it. All right, that's why we find it under the epithelia. So loose connective tissue I usually find under the epithelia, and the major example of loose connective tissue found there is what? Areola. This is reticular, found in the spleen, thymus, and bone marrow. Extremely important. And this is what the dense regular connective tissue. See how smooth it is, how regular it is. Very important. This is dense irregular, looking catastrophic. This is chaos. All right. Now we talk about the adipose as well. We have white and brown fat. I'm jumping it. Don't want to spend time on it. We have the cartilage. Now, for this thing, the major thing I need you guys to know is that anytime we talk about sites are things that are trapped in lacuna. Chondrocytes, osteocytes. Think of trapping, think of lacuna. All right. Chondroblasts are cartilage cells that produce the matrix. So chondroblasts, they produce the matrix. This is the matrix. The same thing with the osteoblasts. They create the matrix. Now, some of the chondroblasts or osteoblasts that are trapped in that matrix are called what? Chondrocytes. Remember that. Know it and know peace, people. My people. Know it and know peace. This is the hyaline cartilage. All right. This is elastic cartilage. Where do we usually find elastic cartilage? External ear and epiglottis. They always ask this question. External ear, elastic cartilage. All right. Then this is fibrocartilage. We find it in the pubis symphysis, in the meniscus, and also in the intervertebral disc. Extremely important. All right. Now for bone, we have spongy and compact bone. Spongy and compact bone. We will discuss this when we are talking about bone. This bone looks like onions. All right. If you see this diagram for your lab, you definitely have to know this answer. This is onion skinning. This is compact bone. Now, you see what I told you about osteocytes? They are mature bone cells that are trapped in where? The lacuna. So if you see trapping, lacuna, you're talking about what? Sites, osteocytes. These are the blood, this thing. Remember, blood is also a connective tissue, specialized connective tissue. Bone is a specialized connective tissue, all right? Now, let's jump to muscular tissues, nervous and muscular tissue. Nervous tissue is very excitable, ability to respond to, excitability is the ability to respond to stimuli by changing membrane potential. There is a question about it. Membrane potential is the difference in electricity or electrical charge, all right, within the membrane, all right? We can have these changes in nerve cells, we can have this in muscle cells. They lead to contraction. Now, this is a nervous cell, nervous tissue, very important, specialized for communication by electrical and chemical signal. Then we ask this. Now, the most important thing you need to know here is that the nervous system, the nervous tissue, right, has the what? The neuroglia. The neuroglia and the what? The neurons. Neuroglia and neurons. They will ask you, what is the nervous tissue made of? Neuroglia and what? Neurons. All right? This is the neurosoma. This is the axons, and this is dendrites. I need to hurry, please. Now, we have the muscular tissues. All right? Muscular tissue, what do you need to know? Which one is striated? Which one is voluntary? That's what you need to know. Skeletal has striations and it is voluntary. It has striations. This is striations and it is voluntary. And what is causing the striations? Alternative light and dark bands, and it's voluntary. Cardiac is striated and it is involuntary. But the striation is not alternative light and dark bands. Instead, the striation is what? Is intercalated disc. Remember this. Know this and know peace. Then the cardiac is smooth. See, everybody is swimming. Smooth. Involuntary and what? Lacking what? Striations. That's all you need to know. All right? Then you need to know cell junctions. You need to know tight junctions. You need to know desmosomes and hemidesmosomes and gap junctions. All right? Now, this part is also very important. You need to know exocrine glands. Exocrine glands. Example is sweat and tear glands. Put it as exocrine. Then we have endocrine glands. The difference between endo and exo is just that in exocrine glands, they are close to each other. So they move through ducts. Tears move through ducts, tear ducts, sweat ducts. Anything duct is exocrine. All right. Endocrine uses blood. That's why we talk endocrine. We talk about what? Hormones. Remember this. Know this and know peace. All right. Now, another thing I need you guys to know is these guys here: merocrine, apocrine, and holocrine. Another name for merocrine is what? Ecrine. I will ask you this question, or they will ask you this question. Ecrine is the same thing as merocrine. All right. An example of merocrine is what? Tear glands, pancreatic juice, and what? Gastric glands. Apocrine, anything apo is what? Lipid. That's milk. Then we have the holocrine. All right. Holocrine is oils in oil glands of the scalp. You will see a question about it. And skin. That's holocrine. All right. Then this is membrane. This is diagrams that you need to go study at your leisure. Now, hyperplasia, hypertrophy, and neoplasia. These are the concepts you must know. I'm not going to describe them. Hyperplasia is just multiplication. Hypertrophy is enlargement. If I get fat and fill this my room now, it becomes what? Hypertrophy. People that are gymming, you know, they gym. See me now. You see my hand? See my muscle? This is hypertrophy. Don't laugh. Don't laugh at me. This is hypertrophy. Then hyperplasia is when I give birth to a lot of children here. That's hyperplasia. Neoplasia is what? Tumor. Anything neoplasia, you are talking about tumor. When you are talking about differentiation, you are talking about specialization of stem cells. All right? Then metaplasia, I've already described metaplasia. I'm not talking about it again. Stem cells are undifferentiated. All right? Then we have embryonic stem cells, that's total and plural. Adult stem cells are what? Multi and uni. All right? Now we have regeneration. That's replacement of dead tissues. We have fibrosis. Now, if I cut off this tissue and it replaces by this same tissue, that's regeneration. But if I cut off this tissue and it replaces by what? By scar tissue, that's what we call fibrosis. All right? The two of them are replacements, but it's all about replacement by what? And when it's fibrosis, the function is not restored, but regeneration, the function is what? Restored. All right? These are wound healing. You look it up at your leisure. Yes, you need to know atrophy, shrinking. Atrophy, shrinking. So atrophy is opposite of hypertrophy and hyperplasia. Remember, hypertrophy is increase in size. Hyperplasia is increase by multiplication. Opposite of the two of them is atrophy. Did I say atrophy? Atrophy. All right? Now, necrosis is pathological. Pathological tissue death due to what? Trauma, toxins, and what? Infection. Example of necrosis, we have infarction, we have gangrene. Example of gangrene, we have dry gangrene, wet gangrene, gas gangrene, decubitus ulcer. All right? Then we have apoptosis. Apoptosis is programmed cell death. If a cell has not been programmed to die and something kills that cell, like trauma, like infection, like toxins, it's called what? Necrosis. But if it's a cell that died naturally, physiologically, it is what? It is apoptosis. But if it is one that shrink, it is what? Atrophy. You need to know the differences between these things. And that is it. If you have a question, you drop. And if you don't have a question, we have 31 people here. I applaud you for being with me. All right? Please don't sleep. If you sleep, wake up yourself. Wake yourself up. So I'm going to remove sharing. Wake yourself up, guys. Yeah, this is AP. Okay. Oh, some people have run away. Please don't run away from me. I love you. Don't run away from me. All right. So let me. We are up. Perfect. I will know if you're up when I throw questions and I get up to 20 responses at once. That's when I know that you are truly up and running. Okay. Okay. Good. Perfect. You know why I like you? Because you guys answer very fast and it makes me move very fast. It makes me move with the speed of light. So testing the microphone. Let me see people that are up. Testing the microphone. The 4 primary tissue types in adult organs include all of the following except options: connective tissue, fibrous, nervous, epithelia, muscular. A, connective; B, fibrous; C, nervous; D, epithelia; E, muscular. Let me see how many people are up. A lot of people are up. Oh, I'm impressed. I'm very impressed. Okay, so the answer is 5. This is just testing the microphone. Don't think you know it all. It's just testing the microphone. Just testing the microphone. So 4 primary tissue types: connective, nervous, epithelial, and muscular. Still testing the microphone. Still testing the microphone. The clear gel inside the cell is called dash. Still testing the microphone. Okay, we have A, matrix; B, ground substance; C, interstitial fluid; D, cytosol; and E, tissue fluid. You guys are so correct. It is what? Cytosol. You guys are so correct. So correct. That's this intracellular fluid. Okay, not ground substance, please. Not ground substance. It's cytosol. All right. Look at this question. Just hold on. Let me put the options in the box. This is A. Just relax. Let me put the options in the box. This is B. This is C. This is D. And this is E. Okay, let's see. The dash gives rise to the dash. Let's see who remembers the first page. Okay. One more person. Okay. You guys are so good. Give me a minute. I'm trying to bring out this question. Yes, this particular question. Okay. Yes, you guys are correct. The answer is C. Ectoderm gives rise to nervous system and epidermis. Okay. That's fine. That's fine. I know you guys will conquer that particular question. Now, no, I'm not going to ask this one. Okay, let me see if I can. No, I'm not asking this question. It's too cheap. We don't have that kind of time. It's too cheap. It's too cheap. We are not doing cheap questions. You are awake so that you will see challenging questions, right? So look at this question. What's the answer? Quickly, testing the microphone. Hello. The histological section that divides a specimen along its long axis is called A or and dash section. Bronze is on fire. Somebody is on fire. Somebody is pium. Okay, that's good. It's longitudinal. You guys are correct, eh? You guys are correct. I'll be moving faster, you know. You guys are correct. So give me this answer quickly. It's two answers. The basement membrane is found between dash and dash. Okay, yes, the answer is epithelium and connective tissue. This is a cheap answer. Don't think you are now. I'm just joking. Okay. Now look at this question. Yes, this is the question. Yeah, I finally see this question. Challenging. Okay. Somebody should let me see the first 10 people that gives me the answer to this question. First 10 people. Let's go. Mm-hmm. Let's go. Keep going. It seems it's the same group of 10 people that are still answering very fast too. What is happening? Are they the only ones in this class? Yes, you guys are so correct. That's simple squamous. You guys are so correct. Okay, look at this. Quickly. Most kidney tubules are made of dash epithelial tissue, which is specialized for absorption and secretion. Oh, some people are still in the other one. Yes, the other one is simple squamous. Yeah, the other one is simple squamous. So what's the answer to this one? Most kidney tubules are made up of what? Keep going. Yes, most of them are made up of what? Simple cuboidal epithelium. Okay, that's good. Look at this. I told you simple cuboidal produces fluid. Yes. Yeah, you're correct. Look at this one. Quickly. You have to give me two answers in one. Dash epithelium provides a moist and slippery surface and is well suited to resist stress as seen in the dash. Do I have to give you options? Okay. Options A, stratified tongue, keratinized tongue, non-keratinized skin, keratinized skin, non-keratinized vagina. I'll give the options again. Yes, I'll give the options again. A, stratified tongue. B, stratified tongue. C, non-keratinized tongue. D, keratinized. Okay. Yeah. Yeah. Okay. You guys are so correct. The answer is E, non-keratinized vagina. Non-keratinized what? Vagina. That's the answer. Okay. You guys are challenging me to bring out question from archive. You guys are challenging me to bring out question from archive. Okay. An epithelium whose cells are tall and narrow. Once you see this tall and narrow, you should even be able to tell the answer. But that's not all. You need to tell the answer. With each one touching the basement membrane and reaching the apical surface. You see what I was telling you? Each one, touching the basement membrane and reaching the apical surface. Let me see. Keep going. Let's have it. You guys are so perfect. The answer is what? Simple columnar. Thank you so much. Some people can mistake it from pseudostratified, and that option, the first one, is pseudostratified. Don't allow them confuse you. Okay, give me the answer to this. It's two answers. Dash epithelium is the most widespread epithelium in the body, whereas dash epithelium is rare. I'm not giving option. Oh, you think I'm giving option? Nah, no options for you. Dash epithelium is the most widespread in the body, whereas dash epithelium is rare. Let me see. Somebody says stratified squamous, and somebody says stratified columnar. Stratified squamous. If you're writing squamous, you need to be specific. Is it simple or stratified? Stratified squamous, stratified columnar. Okay. Stratified squamous, stratified. Okay, keep going. Yes, you guys are so correct. The answer is stratified squamous. Stratified squamous, like the skin, it's everywhere. All right. They didn't put keratinized, but just think about it that direction. And the stratified columna is so rare. Have you ever heard stratified columna? Look at this technical question. Yes. I love. Oh, sorry. Look at this technical question, guys. A brush border of microvilli is found in dash epithelium, which can be found in the dash. Okay, options: pseudostratified nasal cavity, simple cuboidal esophagus, simple columnar small intestine, stratified squamous anal canal, stratified squamous esophagus. Answer. Okay. Okay, good. I hope you guys are engaged and active. All right. If you have not clicked the like, click it. Okay, somebody's sending me love. You too. How do they do it? This is how they put it. This is my heart, right? Yes. Yes, you guys are correct. The answer is what? Simple columnar and small intestine. When you talk about microvilli of the brush border enzymes are the enzymes in the small intestine for absorption. All right, brush border. You understand? You see microvilli there. So they are talking about simple columnar. That's the intestines. Simple columnar, stomach, simple columnar. All right. Yeah. So that's the answer. Let me see. I want to show you that there is no big deal, in this, because somebody told me that he has a problem with tissues. You see how you guys are answering this in very fast. Let me see the first 10 people. Move it. First 10 people. All cells in dash epithelium reach the basement membrane, but only cells that reach the free surface have cilia. Answer. Ha. Somebody is very hot this night, too. Somebody is killing it. Keep going. Keep going. Yes, you guys are so correct. It's pseudostratified epithelium. I love the fact that you guys are so correct. So look at this question. This is the question. Let me give you the options. This is A. This is B. This is C. Somebody has already started answering. Okay, so let me stop giving options now. Okay, I'll give you only 3 options. A, B, C. Found in the urinary bladder. Dash resembles dash, but the apical surfaces are rounded, not flattened. Answer. Finally, I'm seeing questions that are conquering them. Before, by now, more than 20 people have answered. But you see, now it's just 5 people. Okay. It's getting tougher. They're getting tougher. Yeah, the urothelium is transitional. Don't let them confuse you with the urothelium. Urothelium. It's transitional. Yes, the answer is A, all right? The answer is A, is urothelium, because they resemble stratified squamous epithelium. Don't get yourself confused, all right? Urothelium and transitional, the same thing. Look at this question. Very fast, guys. Very fast. Give me the answer. Very fast. Some simple columnar and pseudostratified columnar epithelia have dash cells. And that cell that they have produces a protective mucus coating over the... Hi, Jesus. These people are answering so fast, though. Okay, perfect. You guys are the people. Uh-huh. I'm feeling you guys, man. I'm feeling you guys. Keep going, keep going, keep going. If you don't know it, right, not sure. But everybody has to type. We have more than 30 people here. Everybody must be typing. Even if somebody has typed that your answer, don't say, okay, that's my answer. No, type. Thank you. Okay, perfect. You guys are so correct. That is goblet cells, all right? Why I wanted everybody to type it is that if you see something similar in the exam, the fact that you type that answer will make you remember it. All right. So don't just wait, okay, it's goblet. You may not see it in the exam and you may not even remember. You see something similar. Right? But when you type it, your brain has already, taken account of that particular answer. Okay, so look at this question. Dash is associated with rapid transportation of substances through a membrane, whereas dash is associated with resistance to abrasion. A, simple columnar epithelium urothelium, that's A. B, simple squamous, stratified squamous. C, simple cuboidal pseudostratified. D, pseudostratified simple squamous. E, pseudostratified simple cuboidal. Not sure. Okay, that's good. Someone said 911. Which are okay. That question, okay, good. I will talk about it. So the question says dash is associated with rapid transport of substances through membrane. So when they talk about rapid transport of substances through membrane, you think of something like alveoli, where air, move past the membrane. So you need to be very light. Remember, simple squamous is the lightest of them all. Like when I talk about lights, I'm talking about this ticket that they gave me. They gave me tickets. So I'm talking about this kind of paper. You see how light this paper is. That's this. You see how light it is. That's simple squamous. All right. Now, whereas dash is associated with resistance to abrasion. Resistant to abrasion. Remember when I told you about the vagina? All right. For example, the vagina is usually simple cuboidal, but because of that abrasion, because when the penis is going in and out, there's abrasion. That's why there are some children that like maybe they are raped. You understand? That's how we are able to detect it, because it will injure them a lot, unlike adults that have consensual sex. Do you understand? So that's what you need to remember. If you remember it, you'll be good to go. All right. When you think about abrasion. Okay. Oh my God. I got tickets for the first time this week. Really? First time? Ha. Me. I did subscription with New York tickets this thing every month. There must be one sent to my house. It's a subscription for me. So I've given up trying to avoid it. Okay. So incoming. Incoming. See, I'm stimulating the economy. Please, I don't want to be stimulating any economy. Please, 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 please. I'm stimulating an economy and I'm depleting my own economy. Hey, do you know if I tell you how much I spend on tickets every month? And I don't understand how. I don't even run fast. I'm not a speedy. I don't know. It's just, there's a spirit that pushes my vehicle fast anytime it sees camera. It loves, I'm telling you, it just, my car, you know the way, some people like to be snapped, picture. They will, every time they see a camera, they pose. That's how my car is. My car loves to be snapped. It just loves it. Like, I don't understand though. It has to be village people. It has to be. It must be village people. They got visa and came to the US. Please don't sleep on me. I'm sorry. I know I can tell stories a lot. Okay, look at this question, guys. You got to put your car on a cruise control. I'll just go and buy a Tesla. That's what I will do. Save money to them. I'll just buy Tesla. And the thing will just drive itself. I'll just leave it auto. Yeah, I cannot come and stress myself. Oh. Let me see. If it now gets ticket now, then that's where both of us will have a problem. So what's the answer to that, guys? What's the answer? Lacuna. Perfect. Okay. So let's speed up. Let's speed up. Some people need to sleep. Let's speed up. Look at this question. Look at this question. Please, I'll put this question, then I'll put the options. Just give me a moment. Let me put the options. Yes, that answer is lacuna, all right? It's a giveaway. That's why I'm not talking about it. Your car needs an exhaust. I'm telling you, my friend. I'm telling you. There's a problem with that car. It just likes to get snapped. Who does that? Snaps more pictures than myself. So that's A. This is B. This is C. Somebody's ahead of us. Oh. This is D. And this is E. Okay, answer. A, B, C, D, E. Collagenous fibers are very abundant in oh yeah, this question. Hurry up, hurry up. I need to post this question. It's very important. Move it, guys. We have 30 something people here. I need up to 20 people answering. It's just some group of 7 people that are answering all this while. Come on, let's move it, guys. Perfect. You guys are so correct. The answer is D. Okay, look at this. Somebody is just excited. A connective tissue fiber refers to A or an dash. A nerve fiber refers to a dash. And a muscle fiber is a dash. All right. So look at the options. This is A. Hold on. Let me give you the options. That's A. This is B. This is C. This is D. And this is E. Okay, so the fact that you guys are now quiet means that this question is difficult. Ha. 4, 5, 6, 7, 8. None of them got it. Yes, you guys are correct. It's E. Complex of macromolecules, part of a cell. Nerve fiber is part of a cell. A muscle fiber is an entire cell. Yeah. Okay, why E? Yes. Connective tissue fibers refers to macromolecules. If you see connective tissue, right, there's a lot of macromolecules in it. Like you see its fiber, they are macromolecule. But if you are talking about the nerve, you understand, a nerve cell, a nerve fiber, you understand, is just a part of the nerve. But a muscle fiber is an entire cell. Do you understand? Like when you do muscle, you understand what I'm talking about. If you bring out a muscle fiber, that muscle fiber is an entire muscle cell. That's what they're telling you. All right. But nerve fiber is not an entire nerve cell. Nerve fiber is just a part of the nerve cell. All right. So that's why that's the answer. And if you look at the other options, for example, they say tissue, connective tissue fiber, entire cell. You know, no. When you're talking about entire cell, you're talking about what? Muscle fiber. So that's the thing. Now, there's a question on Marfan syndrome, but no, you don't need Marfan syndrome because that is pathology. Instead, there is this question that you need. All of you need. Look at this question. Okay, I see you. Perez. Look at this question. Which of the following does not account for gelatinous consistency of connective tissue gland substance? Does not. Does not. So look at the options. Let me give you the options. So pay attention. Does not. A. Glycosaminoglycans, GAGs. B. Proteoglycans. C. Chondroitin sulfate. D. Hyaluronic acid. And E. Collagen. GAG, proteoglycans, chondroitin sulfate, hyaluronic acid, collagen. Answer. And they got it to it's collagen. You are right. All of you are correct. Because all the other ones are, they help in that consistency, right? Glycosaminoglycans, I told you, example is chondroitin sulfates. We have the hyaluronic acid, then we have chondroitin. We have also proteoglycans. So collagen is not part of it, all right? So you guys are correct. Look at this question, please. This question is extremely interesting. Look at the question. Glycosaminoglycans, that's GAGs, help give the gland substance some of its qualities, including dash. What is the quality? A. Hold on. I'll put option here. I'll put the options so that you get it. I know you guys will get this question. I know. Just I shouldn't be wasting my time bringing this particular one. But it's okay. The Lord is my shepherd. B. C. D. Okay, choose your answer. Karina, if you want to sleep, do like this, do yourself like this. All right. Second. Yes, you guys are correct. So I'll be moving with the speed of light. There are some things I really need you guys to know before I jump into skin. So yes, the answer is correct. That's B, all right? Holding water. That's B. Look at this question, guys. An example of highly vascular tissue is dash. An example of tissue that is not highly vascular is dash. This one, you cannot fail it. Options: epithelial cartilage. B. Muscular osseous. C. Dense connective tissue, dense irregular connective tissue. Dense regular and dense irregular. D. Areolar cartilage. E. Nervous osseous. Exactly. You guys are correct. Areola, the most vascularized cartilage, the least vascularized. Sometimes it does not even have any vascularization at all, right? Okay. Look at this question very fast, guys, very fast. I'm trying to speed up very fast. We already spent 2 hours, 21 minutes. That's a lot. And I hope that all of you passes your exams so well. That is. Yeah, options. Okay, let me give the options, please. Dense regular connective tissue A, dense irregular B, elastic cartilage C, fibrocartilage D, ligaments E. You're welcome. Answer, elastic cartilage. Please, it's elastic cartilage. Don't choose something else. It's elastic cartilage. Now, there's function of blood. All of you know function of blood, so I'm not talking about it. I'm not talking about it. Ha, this question is very, very funny. It's very, the answer is even funny, more funny than the question. I'll give you the answer. Look at this question. Which of the following, yes, that is elastic tissue. See, yeah, you guys are correct. Which of the following is not a category of connective tissue? Which is not category? A, no. I'll give this, I'll put it in the box. This A, so that you laugh with me. I know you will laugh. You'll be like, oh, what stupid question is this? This B, C, D, E. Answer. Quickly. Which of the following is not a category of connective tissue? Quickly. Answer. Yes, you guys are so correct. Aerobic. I don't know what aerobic is. I don't know what they are talking about there. So it's not a part, all right? Everything is. Even if you don't know the others, once you just see the aerobic, you should smile and pick it. So look at this question. Nervous tissue consists predominantly of 2 cell types, neurons and dash. What is the other one? Neurons and what? Neurons and what? Hurry up. I just need 5 people. Then yes. Hurry up, guys. Neuroglia. Yeah, the answer is neuroglia. Okay. So look at this question. Quickly. Yes, neuroglia. Answer that other question quickly. Neurons usually have multiple short branch processes called what? Yes, it's neuroglia. I agree. I agree with you. Short processes called what? It has to be 2 answers. Somebody's putting 1 answer. It has to be 2 answers. Oh, wow. Please let me give you the options. A, nerve fibers dendrites. B, nerve fibers glial cells. C, neuroglia cell body. D, dendrite cell body. E, axons dendrites. You guys have been getting it on. Now, it has changed. I think you guys are now so sleepy that you get it wrong. The answer is D, guys. The answer is dendrites and cell body. Where do they receive the information and where is it conducted? It's a cell body. Guys, it's cell body. All right. Yes, you guys flipped it. So look at this question. Astronauts in gravity are able to move food through their digestive tracts because, please, I'll put in the option and I'll put in the option. So just pay attention, please. Because this is A. Anytime I see this tongue emoji, I always laugh. This is B. This is C. Okay, just ABC since people are already answering. So no need of me stressing my fine hands. Okay, you guys are correct. They are able to move food through their digestive tracts because cardiac muscle maintains a high pressure that moves material through the digestive tract. Yes. Sorry, because smooth muscle produces waves of contraction that propel material through the digestive system. What am I saying? So the answer is A. You're correct. Please, I'm not. Okay, fine. I'll just give this question, but you have to answer it very fast. You have to answer it very, very fast. Please, quickly. Aha. I see a question. Aha. I see a question. Aha. So cardiac muscle is described as what? Striated and skeletal muscle is described as who said involuntary there? Okay, striated and voluntary. Okay, look at this question, guys. I want to put the options quickly. That's it. I want to put the options quickly. That's the question. I want you to read the questions in your head quickly. Your exams are usually time. It's time that some people know what they answer, but they waste too much time on questions. So I need you to look at the question and get it. This B. This is C. This is D. This is E. Interesting. Okay. This question has conquered you guys. Only one person has even made a comment. This question is conquering you guys. Not sure. For the first time, bronze is not sure. Interesting. We did see a question on the test. If yes, 911. Hey, I don't know, my dear. 911 is finally. Let me raise my shoulders. Finally. Okay, so listen. Now, that question said, how would the genetic inheritance of a gene producing abdominal gap junctions affect muscle tissue? I want to tell you guys now, you have to put it in your head. That's how you conquer the 911. Put it in your head. Put it in your head that skeletal muscle does not have gap junction. I don't know if you know this information before. Skeletal muscle does not have what? Gap junction. The only muscles that have gap junctions are cardiac muscle and smooth muscle. Skeletal does not have gap junction. So I want you to relate it to the question and give me the answer. How would the genetic material inheritance of a gene producing abnormal gap junction? If it is abnormal, it can only affect the muscles, the muscle cells that has gap junction. I don't know if it makes sense. Yes. Yes, specifically skeletal muscle, which is voluntary. Yeah, you're correct. So the answer is what? Okay, everybody put in your answer again. I'm giving you a second choice, second chance. How would the genetic inheritance of a gene producing abnormal gap junction affect muscle cells? So skeletal muscle does not have, gap junction. So will it be affected? That's the question. No, it will not be affected. The ones that will be affected are the ones that have what? Gap junction. Like cardiac muscle, like smooth muscle. So if you look at that C, exactly that's why C is the answer. If you look at that C, cardiac muscle and some smooth muscle would function what? Abnormally because they have gap junction. But skeletal muscle will be unaffected because it does not have gap junction. Chicken out. Chicken out, chicken out, chicken out, chicken out, chicken out, chicken out. Okay, at least I got you people today. Okay, look at this. Look at this. Options A is 3 questions. So relax. Let me put the options. Is A. I said C. Oh, you said C? Yes, I see it. You did well. Someone said I love the dance. Where do you get your energy from? I taught the other class for 3 hours. I don't know, like maybe I'm just hyper naturally. I think I'm just hyper. No, hold on. Let me put the options for you. So I've given A, I've given B, dash, I've given B, then this is C. Yeah, this is C. I think I'm just hyper. And funny enough, I didn't even take anything. I'm just drinking water. This is D and this is E. Pepper soup now. I ate salad. I had salad today. In the morning, I ate oats. That's all. That's the only thing I ate today. So, what is the answer, guys? I need you to tell me the answer, guys. Somebody said C. So, dash secrete dash into dash. A. Goblet cells, mucus, the blood. B. Endocrine glands, hormones, ducts. C. Exocrine glands, hormones, ducts. D. Endocrine glands, hormones, the blood. Answer. Seems this one is confusing you guys. So I need you guys to think about it. Goblet cells secrete mucus, right? Correct? Into the blood. Does it secrete mucus into the blood? No. A is out. Look at B. Endocrine glands secrete hormones. Yes. Into duct? No. B is wrong. Look at C. Exocrine glands secrete hormones. Wrong. Exocrine does not secrete hormones. Look at D. Endocrine glands secrete hormones. Yes. Into the blood. Yes. The answer is D. Exocrine is like sweat, so it's C. So the answer is D, guys. The answer is D, not C. The answer is D. I just analyzed it now. For C, some people say C. C is exocrine glands secrete hormones. It does not secrete hormones. So it made it wrong automatically. Then E, exocrine glands secretes mucus. No. Into the blood. No. Okay. I think 2 more questions and I'm done. Two more questions and I'm done. Yeah, I put the wrong letter. Yes. Okay, 2 more questions and I'm done. This question looks too long. Why? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, look at this question, guys. Quickly. Give me the answer quickly. Quickly. Let me see how fast you can. Give me this answer. Immediately after this tissue, I jump into skin. And the skin, I'm not explaining it. I'll just give you, maybe we'll do just 10 questions on, or 50 questions on skin, and that's it. All right? So what's the answer, guys? Supposed to be two. Merocrine, holocrine. Merocrine, holocrine. Another for merocrine is what? Ecrine, right? So if you see ecrine in you should know that they're also talking about merocrine. So you guys are correct. You guys are wonderful. It's merocrine, holocrine, not exocrine. All right? You guys are so correct. Look at this question. Yeah, it's mero holo. Yeah, or ecrine holo. Any of them can be the answer. Look at this question, guys. Very fast, very fast. So I'm asking you guys the last question for tissue. You guys have conquered me in tissue. Yes, that answer is hypertrophy. Thank you so much. So look at this one. This one is that's the last question for tissue, please. I'm not asking you another question on tissue. You guys have conquered me. I give it to you guys. You guys are good. Yeah, I know some people want to sleep, but if you can just relax for the next 10 to 15 minutes, we'll be done. All right. I'll try to be very fast with this skin. I'm not explaining anything, please. If you want to read it, you can go read your slides or your textbook or whatever works for you. Yes, you guys are so correct. The answer is metaplasia. I need everybody to. I'm jumping into skin now, guys. So no rest for you. Jumping into skin, which is not part of the skin. This one, let me see. Why did this thing join together? I don't understand it. So which of the following is not part of the skin? Epidermis, papillary layer, hypodermis, stratum basal, dermis. Answer. Keep going, keep going. Come on, I have 30 people here. I need at least 15. I know you're tired, but I'm sorry, but 15. So what is the how am I getting energy? Keep going, guys. Yeah, love you too. Love you too. So it's hypodermis. Yes. The answer is hypodermis, all right? I hope you don't need explanation for it. Look at this. Look at this. Which of the following is not found in the epidermis? I'll give you the options. I know it will still join together. Let me see if I can separate them. melanocytes, fibroblasts, stem cells, still joined together. Okay, so we have melanocytes, fibroblasts, stem cells, keratinocytes, tactile cells. Ah, Jesus Christ. These people are, you guys are too hot too. I don't understand. I don't understand. Yes, it's fibroblast. Fibroblast is the odd man out, all right? Look at this question. Look at this question. The skin covering the dash has skin covering the dash. Yeah. Oh wow. Fingertips, palm soles, and blah blah blah. Okay, yeah, you're correct. You're good. You're good. Okay, that's good. That's good. That's good. Oh, okay. Look at this question and give me your answer quickly. Give me your answer quickly. In which of the following regions would a subcutaneous injection be administered? We have stratum basal, stratum spinosum, conium, dermis, hypodermis. Quickly. Yes, the other one is fingertips. Yeah, because of it's in the palms. Fingertips. Yes. So, where do you put your subcutaneous injection? Okay, yeah, you guys are all correct. It's hypodermis. Look at this question. What type of tissue is found in the most superficial layer of the skin? Simple squamous epithelium, stratified squamous epithelium, dense irregular connective tissue, areolar connective tissue, dense regular connective tissue. Answer. Keep going. Where is everybody? Are we sleeping? 10 minutes and we'll be done. Stratified squamous. Perfect. Look at this question. Looking at a slide of thin skin under the microscope, you know that the stratum basal is the dashed layer of the epidermis from the surface. Second, third, first, fifth, fourth. Answer. Looking at the slide of thin skin under the microscope, you know that the stratum basal the answer is fourth, not fifth. I caught you guys. Have you forgotten that this is thin skin? Thin skin does not have what? Lucidium. This is thin skin. This is thin skin, so it's fourth, not fifth. Oh, I caught you. Finally. Some people said third. No. Okay, look at this question. Which of the following cells is largely responsible for the skin color? No, everybody will get this one. Not worried about this one. Okay, keep going. I really like the way you guys are so active. Melanin popping. Melanocytes. You guys are correct. It's melanocytes. All right, give me the answer to this question. I'm flying now. Give me the answer to this question. The fastest rate of mitosis happens where? Yes, the answer is Basel. You guys are very correct. Look at this question quickly. Wow, we spent 2 hours 47 minutes. Whoa. Answer. Perfect. You guys are so good. Yes, it's lucidium. Yes. Sorry, I'm being faster now. Which layer is seen only in the thick skin? Oh, I've given this question before. That's lucidium. Sorry. Thought I copied the other. Look at this one. The hypodermis is characterized by abundance of which tissue? Which tissue is in the hypodermis? Oh, this question is interesting. Hurry up, give me the answer. Which tissue? Which tissue? Keep going. Bronze, which type of connective tissue? Okay, you guys are so correct. The answer is adipose. You're correct. Now look at this question. Quickly. Adi, yes. The skin discoloration most likely to suggest physical abuse is what? Let me give you the options. Yes, exactly. Look at the options. Pallor, albinism, erythema, hematoma, jaundice. I don't know why this thing is not separating. Hematoma, jaundice. Which one? Exactly. The answer is hematoma. Look at this. A dash is an elevated patch of melanized skin. Answer. You guys are on fire. Trust, honestly. Yes, it's mole. You are correct. You guys are on fire this night, man. Oh yeah, look at this. Not bad. Yeah, this is A. I'll give you just 3 options, please, because we are in a hurry. This is B. This is C. Let me see. Anatelekata. Anakatatele. Okay, good. I'm now seeing contrasting option answers. That's good. A, B, A, B, A, B, A. Action. Action. So the answer is action. ACT, act. ACT, anagen, catagen, and telogen, guys. It's ACT. ACT. Mm. It's ACT, acts. Hold on, that should say from. That's my slide. Okay. I think this might be my last question for you. My last question for you. Yes, this should be my last question for you. Told you I'm not giving. This one is melanoma. No, I don't. As I said, carcinoma. This is bones. No. 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 Think. Yeah, just hold on. I want to be sure that I got you guys covered. Okay, you guys have answered. Everybody didn't answer, but that's fine. The answer is apocrine glands. And guys, this is the last question I have for you. I've tried, right? We spent 2 hours, 54 minutes. Jesus. And why is it looking like I'm not even tired? You will see it now. For people that are in my class, you know what I'm always doing now. You know my direction and dimension. You know. Thank you so much. And you guys also did well, honestly, because like you guys were here with me for almost 3 hours. I planned this class to be just 2 hours, but this is 3 hours. But what I need you guys to do is to, just do your little review. Yeah. For people that are writing exam tomorrow, write your exam tomorrow. People are writing exam with other professors, write your exam. And I believe that you will do well. You're welcome. I believe that you guys will do well. All right. What you need to do now, just go and sleep. Sleep. Then in the morning, maybe you can now do your revision. But we've already done the revision for the 3 topics. All right. Now, if you write your exam and it helps you, you can come back on the channel to talk about it. And also we can also discuss it later to know if we can continue this, like before every exam, midterm, final exam, everything. We bring out time like this to do it. All right. Thank you guys very much. Good night. Take care of yourself. It's nice to have you guys. Yes, good night. Take care. We do this before every exam. No, but you need to write your exam first and know if it helps you. If it helps you, then maybe we can be doing it because you can't be sure. Yes, good night. Take care. Yeah, exactly. Exactly. If it's effective because This is the first time I'm doing this kind of revision with students before exam, and I just want to see what works for you. You know, what helps you in exam. The most for me, it's all about you guys smashing your exams. All right, no matter the exam, you know, at all, you know, we could always go fix a class for it. And I always like the classes to be immediately before the exam so that it's really fresh in your head. So even if you sleep and you have an exam tomorrow morning and you sleep and you don't study anything, if you come to the exam hall tomorrow and you recognize some of the, you know, the similar questions, you'll be able to dust it if you were following us from beginning to the ending. All right. So good night, everybody. Take care. Yeah. Bye bye.